All right, let's review your Chapter 13 study guide for your test at the end of this upcoming week. So number one, what does RNA have that DNA does not have? And what is the sugar molecule in RNA? Well, RNA has uracil. RNA contains the letters A, C, G, and U, and DNA contains A, C, G, and T. So the difference you can see is between this T and this U, and that U is uracil. And that's what RNA has that DNA does not have. And the sugar molecule in RNA, that is going to be ribose. Number two, what type of RNA carries information from the DNA to the outside of the nucleus? Now remember, DNA is found on the inside of the nucleus, and we extracted DNA, and DNA can fit inside a nucleus, but it can't go outside the nucleus because it's too large. And the nucleus has nuclear pores. That's small enough for the mRNA to come in and make a copy of the DNA. So the mRNA comes into the nucleus, makes a copy of the DNA, exits the nucleus into the cytoplasm, and we'll move to the ribosome. We said ribosomes make proteins. Now you know why. And the mRNA will make its way to the ribosome. This is also called transcription. So when mRNA is making a copy of DNA, it's called transcription. Number three, what is mRNA transcribed from? So where does mRNA come from? And the reason we said that is transcription is because we have this question right here. What is mRNA transcribed from? In other words, where does it come from? And it makes a copy of the DNA. So that's where mRNA comes from. All right, moving on. Think about amino acids. Amino acids are the building blocks of proteins. How many letters do we need to create an amino acid? And what do we call these three letters? Well, the three letters, we have to have three letters to use the chart, that big circle wheel chart that we looked at to find those amino acids. When we call these three letters codons. Number five, what happens during transcription? Is this DNA to mRNA or is this mRNA to tRNA? Now, we've already talked about transcription, what happens during being transcribed. So this one you should be familiar with, and that's DNA to mRNA. Just so you know, this is correct. It should be a lower capital M, and that stands for messenger RNA, which we'll talk about later on. That brings us to number six. What happens during translation? Well, translation happens next. And if you can remember that DNA to mRNA is transcription, then that means mRNA to tRNA will be translation. Number seven, what does mRNA mean and tRNA mean? So we have that little m and that little t. What exactly does that stand for? Well, the mRNA stands for messenger RNA, and the tRNA stands for transfer RNA. Number eight, what is the general sequence to make protein? So we talk about protein synthesis. What is the general sequence? Well, it's DNA to RNA to proteins. So mRNA and tRNA go in the RNA section because mRNA comes in, makes a copy of the DNA. The mRNA is complementary to the tRNA, and then the tRNA will bring in amino acids based on the mRNA, and amino acids are used to make proteins. Number nine, the genetic code, the bases, the A, the C, the G, the T, and the U, T being DNA, U being RNA, they're always read how? And that's going to be three bases, which are the letters, and they're always going to be read three letters at a time from left to right. There are no exceptions to that rule. Number 10, a mutation that involves one or just a few nucleotides is called what? And that's called a point mutation. An example of a point mutation is substitution. We looked in class how a letter A got changed out for the letter G. And that happens at one point or a few nucleotides, but not together, just somewhere along the DNA. It can happen together to two letters in a row, but the letters aren't being added or deleted. We are just changing one letter out for another. So we're not adding letters or deleting letters. We're just making a substitution. Number 11, what are the four types of chromosomal mutations? So this is what we looked at in class. So here, these are not codons. I know these are not codons because we have A, B, C, D, E, F, and the letters for codons are A, C, G, and T, or if you're looking at the mRNA, A, C, G, and U. So these are not codons. You can think of these as sections of information. So here we have A, B, C, D, E, F. On the left side here is what is normal. Over here is the mutation. So over here, I don't have A, B, C. I have A, C, D, E, F. The B has gone missing. It's been deleted, and it is called deletion. pretty easy one to remember. This one, a lot of students mess up, not because they don't know what's going on, they just use the wrong word. We should have A, B, C, D, E, F. Here we have A, B, B, C, D, E, F. The number one mistake students make is saying that this is insertion. It's not insertion because we're not inserting something new. We are copying something that is already there. The B is being copied, and this is what we call duplication. Now over here we have A, B, C, D, E, F. And we don't have BC at the beginning, we have it at the end. We don't have DE at the end, we have it at the beginning. And notice they've been flipped, they are backwards. It doesn't go DE, it goes ED. This goes CB. Because it's been flipped, we call this inversion. And this last one here is really easy. It's the only chromosomal mutation that has 2 chromosomes that are easily identifiable. This one does too. It looks like a pretzel. This one is spread out a little more. It's a little easier to see. Here we have ABC, but the DEF is down here. This chromosome has picked up information from the second one, and the second chromosome has picked up information from the first one. Because they have changed location, we call this translocation. All right, number 12, what happens when a letter is deleted from a base in regards to information? Well, it says in regards to information, and when we take a letter away from a codon, that's going to lose information. You're going to lose part of that information, and we call that a frame shift mutation, which we'll get into later on. Do most mutations have an effect on an organism? A lot of people who have no idea what they're talking about will tell you mutations are deadly. Well, most mutations have no effect on an organism at all. And this is because if you look on that giant circle chart that we have, that we use to find amino acids, you'll notice that more than one codon can code for the exact same thing. Number 14, what does the Lac Operon do? And we're going to cover this in class, but it turns off the protein or gene that is associated with consuming glucose. That's what it does. And operons are the regulators for prokaryotes, which are going to be your bacteria. Number 15, what is involved in the regulation of eukaryotic genes? So we said operons are the regulator for prokaryotes. What's the regulation of eukaryotes? And that's going to be the TATA boxes. What is found only in prokaryotic genes? That's going to be your operons. So 14, 15, and 16 are dealing with prokaryotes and eukaryotes, specifically giving you some background information on how protein synthesis works for them. What are the Hox genes and what do they determine? The Hox genes are really wonderful. They're found in living organisms and they determine your body plan. You can find them in almost every living organism. You can find them in fish. You can find them in flies, birds, humans. You can find them in almost every single living organism. It determines where your eyes go, your arms go, your legs go, and determines where your body parts go. This way you don't have a leg sticking out of your face. So what does RNA polymerase bind to? Well, this is how RNA is made. So it's going to bind to that DNA, making a copy of that DNA. What does the stop codon do? Well, it stops calling amino acids for that protein that is being built. And when it stops calling amino acids for that protein being built, the amino acids that have already been called will be used to build that protein, whatever it may be, based on the mRNA code. Number 20. So what do we call the 3 letters for mRNA and what do we call the 3 letters for tRNA? Well, we've already said this. We said mRNA are codons. Codons are also found in DNA. So when we're talking about codons, we're talking about the 3 letters for mRNA and DNA. And it's any 3 letters. For DNA, it's any combination of A, C, G, and T. It could be AAA, ACG, ATT, TAT, whatever, any combination. For mRNA, because it has a U, any 3 letters from A, C, G, and U, any combination. And for tRNA, they use 3 letters too, but we don't call those codons, we call those anticodons. All right, 21. What is the order of nitrogen bases in DNA determine the order of? So when we're talking about the nitrogen bases, we're talking about the A, C, G, and T, and it's the order of amino acids which will build the proteins. There's only so many amino acids. When you put them in a specific order, it's going to build a different type of protein. All right, 22. The Lac repressor releases the operator in the presence of what? And that is lactose. So what this means is when lactose is detected in the environment, the Lac Operon turns the genes on that control the breakdown of lactose. And lactose breaks down into galactose and glucose because bacteria, they need energy too. And we know glucose is energy. So they're breaking down lactose into two things, galactose and glucose. And this is so, for example, E. coli is going to use glucose, not the galactose. So when E. coli is in the environment, it's going to release the operator when it finds lactose. That way, it can break down the lactose into galactose and glucose so E. coli can use the glucose. Once it's no longer detected, it'll shut that down to preserve energy. All right, number 23. What must happen to a DNA molecule before nucleotides can begin to be assembled into a new RNA strand? Well, it has to be separated into two separate strands because remember, DNA is made up of two strands. RNA has one strand. So if you're going to make a new RNA strand, what you have to do is you need to separate these into individual separate strands. Number 24, what is the central dogma theory of molecular biology? We've already covered this, and it's DNA to RNA to proteins. So the central dogma theory is the basic sequence of protein synthesis or the making of proteins, and we talked about this earlier. So this right here is the central dogma theory of molecular biology. All right, 25. Why are Hox genes so similar even though they are found in various types of organisms? And we said because the Hox gene comes from a common ancestor. And this is why you can find it in almost every living organism. If you pick a random living organism somewhere in the world and you extract its DNA, you will find the Hox gene. If you take our DNA, you will also find the Hox gene. This proves that we all come from a common ancestor. And the last one, number 26, which we've already talked about, just remember that this is deletion, and these are chromosomal mutations, not point mutations. We have duplication, because an item's been duplicated, not inserted. Duplication, not insertion. Inversion, because the chromosome, they have been flipped. And down here, the easiest one, because you can clearly see the two chromosomes, they've changed location, which is translocation. So that's going to be it for that. If you have any questions, you can always ask in class or shoot me a message on Remind. We'll see you guys this week.